My name is Devin Gilson, and in this presentation, I'll be telling you about my examination of cephalopod intelligence. Brief overview of what I'll be going over today is both the introduction to cephalopoda and the definition of intelligence itself, the intelligence shown by cephalopoda and its many occupants and their applications, the genetic IQ puzzle that the genetics of these groups represent, and my concluding thoughts on it. Intelligence is often defined as the total mental capacity of things such as abstract thinking, understanding, communication, and our ability to solve problems. In non-human species, it's much harder to detect abstract thinking and understanding, so we look specifically for their adaptability to new and unusual problems not usually encountered in their native environments. The cephalopoda are a wide varied group, part of the mollusca phylum, and there are three well-known examples: the cuttlefish, as seen in the upper right corner, the octopus, as seen in the upper left corner, and the squid, as seen on the bottom. They have several unique and interesting characteristics, such as their arms, which are lined with suckers that can conform to most shapes and most textures, as well as exert high amounts of force per individual sucker. As an example, the common octopus can exert up to 35 pounds of force per single sucker. Another interesting characteristic is they have the ability to camouflage themselves incredibly well. This ability comes from their special organs called chromatophores. Chromatophores are tiny sacs found in their skin that can expand or contract, revealing tiny dots of color. This allows them to both match and or mimic the color seen around them, in some instances even forming patterns. The other important aspect that we're going to be looking at today are the intelligence displayed by nearly every member of this group. Incorporating the last two, the camouflage and intelligence, we'll be looking at the unique case of the morning cuttlefish. In the example shown here, we see an observational study by Brown et al., in which a male cuttlefish, seen in the middle, labeled M, is displaying a male mating pattern towards the female to his left, labeled F. Off-screen, there is another male who is also vying for the same attention from the female. The male in the middle is showing a deceptive pattern by mimicking the female color pattern towards this aggressive male. This behavior was seen in approximately 40% of observed males, though only in the presence of a single male and a single female. Any more, and it seemed that the cuttlefish's ruse would be discovered by either party and would subsequently be chased off by an angry male. This showed an understanding of the communication system in use and a recognition of the self-awareness of both the female he was courting and the male he was actively deceiving to both con and trick his way into a higher fitness. In this image, we see the setup used by Richter et al. In both A and B, and the data received from his study in graphs A, B, C, and D. The study itself consisted of two interlocking L-shaped containers that an octopus would open for food. There were 4 trial levels. At trial 1, the box was open in a tank with the octopus free to interact with it as it would. In level 1, there was an opaque blue barrier, as seen in image B, in which the octopus could not see through and had a very limited space in which to operate its arms through. In trial 3, the octopus had to flip the L-shaped box over from its normal configuration with the blue opaque barrier still in place. Level 4 consisted of multiple trials in which the alignment and orientation of the L-shaped box was constantly changed between each subsequent test so as not to be too similar to the last one. The average time taken for both of these was graphed in graph B, C, and D, showing the last day of 7 individuals for each trial level as compared to their first trial of the next level up. The graphs B, C, and D show the mean time between the last day, as stated before. In graph A, these are averaged and shown the amount of time taken as the tests are progressing. These graphs in conjunction with A show that while the initial attempt may take longer, the time decreases with repeated exposure, indicating that the animals are showing a developing problem-solving strategy that stays with them through each subsequent test of each level, as opposed to seeing each new trial as an entirely new entity and something to figure out from the very beginning. Here, we begin looking at the genetics and morphological appearances of the octopus's brain and intelligence. In image A, we see the layout of the octopus's internal physiology, namely, the optic lobe, as seen here, the supraesophageal brain, as seen here, and the subesophageal brain, as seen here. Another interesting note is the axial nerve cord, seen going along the arm here. This is found in all of the octopus's 8 arms and contains approximately 300 million of its 550 million neurons. The octopus has roughly 2.7 gigabases to their genome. A human's, by contrast, has approximately 3 gigabases. This shows that the octopus's genome is close, but not quite as large as our own. Of these, I focused on a subgroup of catarrhins, known as protocatarrhins, as seen here, and C2H2 zinc finger proteins, seen here. Protocatarrhins are a superfamily of cell adhesion proteins that play a role in stabilizing multicellular structures. Looking at the heat map in figure B, as seen here, we can see that the Octopus bimaculoides, the first column all the way on the left, has an above-average number of protocatarrhins when compared to some of the other species investigated, including Homo sapiens, as seen in the far right column. These protocatarrhins help to regulate neuronal development and synaptic specificity. In an experiment done on house mouse, also seen in this heat map, here, directly next to Homo sapiens, it explored the results of mutated or missing protocatarrhins. Protocatarrhin alpha, for example, when mutated, caused incomplete axon sorting around the mouse's olfactory bulbs. This resulted in signaling being much more inefficient and much harder to obtain in general. When the protocatarrhin Y genes are absent or otherwise infringed upon, there were neurological defects and even infant death. Also observed a large amount of multiple neuronal subtypes undergoing apoptosis. Higher numbers of these coding areas in octopuses are a possible reason for their adaptive intelligence. The reasoning for this is that the higher amount of neuronal development reduces the risk that the important systems are split up or subject to apoptosis. Also seen in the heat map, as discussed before, are C2H2 genes. These code for zinc finger proteins. These proteins form combinations allowing for complex nucleic acid binding. This aids in creating a stable scaffold for other nucleic acids and transposable elements to attach to, resulting in a more R-adaptive view on gene transcription, where rather than focusing on a few examples of one of these genes, the individual has multiple copies, so as if a few are taken out, the rest can still operate and perform effectively. C2H2s play a vital role in a variety of cellular and genetic activities, such as gene transfer, mRNA trafficking, cytoskeletal organization, protein folding, chromatin binding, and many more. The sheer number of tasks C2H2s are involved in are helping to understand just how they may play an important role in the adaptive strategies that the octopus uses its intelligence for. Some closing regards. 1. A future experiment that I thought of doing during this entire project. Richter et al.'s project could possibly be combined with the house mouse project. To delve deeper, the experiment could look at individual octopi with either of these gene families, C2H2s or protocatarrhins, inhibited or otherwise mutated. Then, put through the similar testing as Richter et al.'s individuals were, in which stages 0, 1, 2, 3, and 4 could be performed by both groups, a control group of healthy individuals, and then the mutated group. Afterwards, their times could be averaged and seen if they were significantly different between the groups, indicating that these genes may play a key role in their product and problem-solving strategies, or if there was no significant difference, pointing us in the direction of something else to study. The information I reviewed seems to point to the fact that these are in fact important in both the neurological and genetic aspects of their intelligence, but I want to stress that I cannot confirm in the absolute, and that further study is of course needed. These are the sources of this project, and thank you for watching.